What is up, Brad fans? How you doing? I hope you're having a good summer. If you saw the episode title and thought, wow, what a hyperbole, what a clickbait title, let me assure you, it is not. We are talking today about a tiny little nematode, which is a small, round, white, insignificant little worm that lives in soil and rotting fruit, as you'll find out, rotting vegetation. But this thing changed biology. It has led to so many discoveries in developmental biology, cancer biology, genetics, the nervous system, so many things, because it became a perfect model system for doing so many of these different types of work. And to help me tell the story of the group of intrepid scientists that set out to basically turn this thing into the great model system that it is, Jonathan Pettit, professor, geneticist at the University of Aberdeen. He joined me to, talk all things C. elegans, as you'll hear. He outlined the history of this model system and of this story, which is a really fascinating one. It's such a science story filled with discovery and serendipity and, just saying, hey, can we do this? Is this possible? Well, let's do it. And it involved building a whole new infrastructure for studying this thing, the C. elegans worm, computing software that needed to be done. There's lasers involved. Very, very fascinating story. And again, a really true testament to science and how it works and the type of people that do it and the types of questions that they ask. We, of course, also talked about science communication, and that conversation went to a place that I wasn't expecting, which was really, really interesting. Jonathan brought up the point of the importance of having a scientifically literate population, especially in genetics, due to the potential ethical quandaries society might have to face. He brought up an example of someone using the defense against a violent crime, that they're genetically predisposed to it. How will we be able to see through that weak argument? And also, we talked about recent events involving violent crime that was racially motivated and potentially inspired by some dubious science. So how do we safeguard against that while having a scientifically literate population? But not as easy as it sounds. So well worth it to stick around to the latter half of the conversation for that bit. Before we get to that, as always, please rate and subscribe the podcast wherever you get your podcasts. Head on over to the website, twobradforyou.wordpress.com. You can follow along there. You can find links to all of the podcast players that you might want to use to rate and subscribe the show. There you will also find all the different ways you can get in touch with the show, our social media links at twobradforyou, Twitter and Instagram, an email, twobradforyou@gmail.com. We have a voicemail box there. Anything you want to say, we want to hear it. We'd love to hear your feedback, and we'd love to play some of your comments on the show. So please do get in touch. And again, please do rate, subscribe, tell your friends about the show. All that stuff helps. And finally, if you feel like donating, there is a link to donate to our Buy Me a Coffee page as well. Thank you so much for listening. And without further delay, here is my conversation with Jonathan Pettit. Well, Jonathan, thank you so much for taking the time. And I guess we should thank John Gilliard for connecting us. So he recommended that I have you on. So thanks, John. And yeah, I think a good place to start. We're going to be talking about C. elegans. I'll let you do the full pronunciation of the Latin name, so I don't butcher it. A tiny little worm. But maybe let's just start. You can give us sort of a brief, like what you do, without giving too much away about the worm, and then we'll jump into the history of this like pretty incredible little worm.
Yeah. So in terms of the pronunciation, I've heard different pronunciations. I'm not an expert on Latin, but I call it Caenorhabditis elegans, and a lot of people I know I learned it from them. So
that's what I've heard too. So
so what we do in the Aberdeen Worm Lab is we're studying something that's specific to C. elegans and in fact in nematodes in general. So we study a process that is required for the flow of information from DNA through to the proteins that make up the cell. And the nematodes use a special trick called splice leader transplicing. It's the technical name, it's not important. What is important is it's specific to nematodes. So our research is aimed at understanding that mechanism so that we can potentially develop drugs that can be used to treat nematode infections. And the advantage of that is that this process doesn't exist in humans or in fact, any mammals or plants. So it's potentially a good drug target.
Mm-hmm. Yeah, so that's kind of like in one of the last episodes I did talking with John, we talked about these parasitic nematodes. So that's the kind of thing you're looking for a target. Yeah, okay. Which is then kind of interesting to then go where we're going to go, which is because we're talking about how this one thing in nematodes isn't found in anything else, but there's actually a lot that is found in everything else from nematodes, and which is one of the reasons C. elegans has become really one of the biggest, most studied model organisms. Can we say that? I think it's fair to say that.
I mean, there's rivalry among the model organisms. A fruit fly geneticist might take issue with that.
Okay, right. Fruit flies is up there.
Good-naturedly, I think. Good-naturedly, yeah.
Right. But then let's talk about, C. elegans and just like what it is. So we're talking about nematodes, little roundworms, but what it is, where it comes from, we'll say, and why this fella named Sidney Brenner thought that this would be the great model organism.
So it's as an adult, C. elegans is about 1 millimeter long. You can see them with the naked eye, but they're challenging. So for the most part, we use microscopes for everything that we do. It's described as cosmopolitan, which I think is a great word. Of course, I always think it sounds very, very. What's the word?
Like worldly
you think
and
of models and
distinguished.
handbags and things like that.
Yeah, exactly.
But actually, it just means that obviously it lives all around the world. And it lives in rotting vegetation. Sometimes you'll see it described as a soil nematode, but I think it's actually much more associated with rotting vegetation because that's where it finds the things that it likes to eat, which is basically bacteria. And it started life as an experimental organism when Sidney Brenner, I think I'm pretty certain he has described himself this way, in that he's a very good starter but not a very good finisher in terms of research projects. So I think he used to get excited about the next best thing. And in the early 60s, he was fresh from the absolute successes of developing molecular biology. He was part of the team that cracked the genetic code. And I think he was a little bit tired of he felt that all of the big questions had been answered in that area. And so he wanted a bigger challenge. And I think at the time he was thinking of the next challenge would be nervous systems. How do brains work? That was the focus. And he said he wanted to quote, tame a metazoan, by which he meant, realistically, he wanted to develop a new model system. And I think he thought fruit flies, which were already well developed, were probably too complex for what he wanted. So he fixed upon a nematode. He initially fixed upon C. briggsii, which is a relative of C. elegans. Can't remember the reasons why, but in the end, C. elegans was developed. And the C. elegans that we use in the laboratory allegedly traces its lineage back to mushroom compost from Bristol in England, because that's where Sidney got his isolate from. And yeah, so that's where that all started. And Sidney and his team in Cambridge at the Laboratory for Molecular Biology then set about developing it as a model organism, which even today would be it's more frequent. Okay, it's easier now with all the technological breakthroughs we have. It's doable for one lab to do. Back then, it was just astonishing the techniques that needed to be put into place in order to do that. And so it's a testament to both their diligence, but also their self-belief, I think. I'm always astounded when I read these early accounts, how confident these guys were that they could do this.
Yeah, I saw that in one of the papers you shared with me. which was sort of a personal account of one of the guys that I think he ended up working on the computer systems or something.
Oh yeah,
with
John
this.
White. Yeah.
Yeah, and this was what came across was he was just kind of enamored with Brenner and how he just seemed like he went around touring around giving a talk about how I'm going to do this. Like, I'm going to make this a model organism and we're going to do it and it's going to be great and all this stuff. But then, like you said, so you got this thing that lives in, rotting plants and stuff. He's got some ideas as to why it might be a great thing to study, which we can get into. But then, just like you said, bringing this thing into the lab, you need a way to keep it alive. You need a way to visualize it, so like microscopes to look at it, all of these ideas as to what you think you might want to do with it. They basically had to build the machines, the housing, the data collection, all of these things in 1970 with what we would consider rudimentary computer systems and all of this stuff. It's like, it is quite incredible.
I mean, Sidney's goal was nothing less than to simulate the nervous system, of a living animal. So he envisaged the idea that they would take electron microscopes, so incredibly magnified images, sections of the worm from head to tail. They knew that the worm, one of the reasons why he chose it was simple. I don't know whether they knew the exact cell number, but they knew it wasn't a lot. It was around about 1,000, and it was John Sulston that nailed that figure down to the specific 959 cells plus the variable number of cells that make the sperm and egg. But they knew that there was a limited number of neurons, and they thought, well, if they could do these sections, basically cutting a sausage from head to tail, because the worm effectively is just a sausage. And then they'd be able to put these things back together in the computer, and they'd see all the connections between the neurons. They'd see the synapses between the neurons. And the goal was, well, let's build a model of that nervous system and in the computer show how the worm works in terms of its behavior.
Right.
That was an impossibly ridiculous goal, even for them. And there are I think there were several PhD students that, I wouldn't say sacrificed their PhDs, but that was an uphill battle, which probably was a little bit overambitious.
Yeah.
And even today, people now are starting to replicate C. elegans behavior. There are Lego robots that can move in ways that C. elegans moves, using effectively the same neuronal architecture, but in silicon.
Right.
So we're getting there, but we're still not there.
Yeah. Yeah.
And that's what I find fascinating. This is an organism that has 300 neurons, about 7,600 synapses. That's tiny.
Yeah.
It's ludicrously small. So Sidney was onto the right thing. It should be tractable experimentally. You should be able to do it. But even today, we're not at that point. Nobody can. There are advances, but nobody's at the point where we can say, there we are. We've got the nervous system for the one. We know exactly how it works.
I mean, it makes sense. And I think in that white paper, he mentioned it like we're going to make like a circuit map, basically, of this organism, which maybe that puts it into some perspective for people. But it is really incredible to think about. Just, like you said, like how ambitious of an idea that is for 1970. Now you can kind of picture it like, oh yeah, we could probably just scan it, and get like a 3D model of something, that's what you envision in your head. But we're talking about like 1970, right? Like, so This is really incredible. So the simplicity of the organism was one of the things that made it, like you said, a right candidate for this kind of project. So we know it only has 950 odd cells.
Yep.
We know that it's 302 neurons. So there's these kind of...
We have to say 300 now.
Oh, okay.
Two of the neurons... I don't think it's it's almost a joke now. It's almost like a meme on Twitter amongst worm scientists that I think it's fairly clear. Two of the cells, it's equivocal whether they're neurons. So we now say 300.
Oh, okay. So just so I don't get shouted at on Twitter by the worm people, we'll say 300. It feels a little bit like Pluto getting,
Right.
designated
I think
not
there
a
is an
planet.
element of that. Yeah.
All right. But I think the point stands. You have these sort
Yes.
of tractable, as you put it, numbers that we can get to. But the other really interesting thing about this that I think is kind of neat and that makes sense is there was transparency. Like you can actually see
Yes.
into the worm. And I think they basically counted the cell divisions. So you start with the egg, the zygote, and then you can just watch it and count how many times it divides, something like this.
Yeah. So this was John Sulston's mammoth project. John Sulston was the person that ultimately led the UK half of the Human Genome Project.
Okay. Yeah.
And I think, and as he said, I'm good at boring and monotonous projects. So he sequenced the worm genome. Having sequenced the worm's genetic code, he felt they were then well placed, and he pitched to the then Wellcome Trust to build an entire centre for sequencing genomes, what became the Sanger Institute now. But going back to the 70s, he was the person that fixed upon the idea, well, I'll just watch I'll watch embryos develop. And that's what he did. So he had 2 microscopes set up in the room. Set up with and he watched this live because there were I think it wasn't at the time when he started, there was no recording mechanism, so recording was simply writing in his notebooks,
Right.
drawing the cells. He had a camera lucida, so it sort of projects images from the microscope as well, so you could draw that way.
Yeah.
And he would move between the 2 microscopes, and I think the whole project was about 5 years.
Wow.
And he wore grooves in the floor as his chair moved between the 2 microscopes. And the outcome of that was the pattern of cell divisions from the first cell, which is the fertilized egg, through to the last cell in the adult. And they were all mapped out in these lovely little diagrams. And the thing that he discovered, well, I'm not sure he discovered because I think people already suspected it, but he nailed it in terms of the detail, was that nematodes have an incredibly fixed pattern of cell divisions. So you can if you know which cell you're looking at, you know exactly what all of its children and grandchildren and great-grandchildren and so forth, as the cells divide, you know exactly what they're going to do in the end. You can't do that for a human embryo. If you were to look at a human embryo, maybe so at the 16-cell stage, you've got no idea generally speaking, what those.
So you mean
cells
like
are.
you could, if you know what cell you're looking at, you could then say, okay, well, the next divisions, these are going to form this cyst,
Yep.
like the intestine or the
Yes.
nerve.
The intestine is a good one. There's a cell in the early embryo which John called E, and E divides to give only intestinal cells.
Incredible.
So in fact, if one of the things that John White, one of the other researchers, developed was a laser, which I think is just amazing, for killing cells. So it's a real James Bond sort of experimental tool. But what you could do is you could ablate, you could kill one of the cells in the early embryo and see what happened.
Right.
And if you kill E, everything else sort of works. The worm develops normally. And it develops into what is a visible worm that hatches, but it has no gut.
So it doesn't last very long.
It doesn't last very long. But these sorts of experiments were really instructive because they could use those to say, well, does the cell need to talk to its neighbors? How important how important is it that it, and at what time is it talking to its neighbors? Again, these are. I mean, a laser is not a simple technique, but its use is quite simple. All you're doing is saying, what happens if this cell is not there? And that's one of the things that why John Sulston's contribution was so important, because he laid the framework for that. And then other researchers come in. So Bob Horvitz, one of the other people that shared the Nobel Prize with John Sulston and Sidney Brenner, he came into the project, was like, okay, so you've got all these cell divisions which happen reproducibly. Well, what's the genetic What's the machinery that makes that happen? And so they looked for mutations. So they randomly mutagenized the worms, and they looked for worms that reproducibly had abnormal cell divisions. And that was the thing that led to an enormous sort of set of breakthroughs, a whole sort of suite of breakthroughs, I suppose. So I guess the two things that C. elegans had, well, it's more than two. You've got the simplicity anyway, the anatomical simplicity, which means that you can measure things at the level of individual cells doing individual cell things, coupled with the genetics. Because one thing we haven't talked about is we grow them on agar plates, we feed them bacteria. It's a very simple system to grow them in. At 20 degrees, they go from an egg to an adult in 3 days. And each worm gives rise to approximately 300 offspring. And so from a geneticist, what we need is short lifespans and lots of offspring. And so it's an almost perfect system for that if you want to study animal biology. Even the Drosophila geneticists, I think, have to concede that in terms of speed of discovery, C. elegans can't really be matched. The only organism that beats that isn't an animal; it's a yeast.
Right, right. I didn't realize we'd be stumbling upon to this thread of fly versus nematode.
Oh, the fly versus nematode thing. I mean, it's a nonsense. It's a nonsense battle. I mean, it doesn't really exist, but it's kind of fun.
Yeah, I could just see genetics conferences being split in the room. It's just no, that's great. But I think this is really cool, and this is like what was so fascinating to me when I worked with John. I didn't work on any of his C. elegans projects that he had going in the lab, or the ivermectin for that matter, but just understanding that you can take this little, like we said, maybe millimeter long organism that you would think, oh, it's just this insignificant thing that eats rotting plants. But now we're seeing this coordinated ballet of cell division to tissues to life to everything that you need. And it's that ability to sort of say, what happens if we remove this? Now that we have this map of like how it goes from egg to adult and all the things that it needs to live, all the basic fundamentals that it needs to live, what happens if we take that out? And you get these questions, like you said, of when does the cell talk to each other? Is it needed to talk to each other? So you have that fundamental thing. You poke at that one question, what happens if I take this out? Which I think is so such a basic scientific question, right? Like, so how much complex science, Nobel Prize winning science is based on, well, what happens if we just take that out? Yeah, something so simple.
Yep. No, that's exactly.
And then you get all these other questions, that brings, and it goes on and on and on. And I think maybe that's a thread of C. elegans as a model organism story, is just the removing something, because you can map it so well. What happens when we then remove something?
Yeah. So, all of this work was part of, the work that was going on in C. elegans was part of a project, a much broader picture, which also includes Drosophila. So about the same time, Drosophila geneticists were asking similar questions, and so you have these two communities of scientists essentially saying, we want to understand how development unfolds from a fertilized egg through to sort of adult tissues. What are the events that must take place? And we know that they're taking place at the level of machines operating in cells, but we don't know what those machines are. We'll break them, and that way we'll find out what they are by literally identifying the genes that have been broken. And it's genuinely pioneering work, because these people were discovering genes that had never been, this is the first time anybody's asked this question. So everything that comes out is new, completely new. And so they're really pioneering the approach. They've got nothing to go on. Today, if you were to do the same project, there's a wealth of data in the databases. So, we've done a little bit of work looking at other nematodes, which haven't yet been studied, and we've got some projects working on other weird and wonderful beasties. But the great thing about it is that if you pull out one of those genes from one of these odd organisms and you put it into a computer to compare it to what's already known, you pull out all this wealth of data. So you know, or you almost know what that gene is doing because it's got all of this data from worms, from yeast, from flies. Back then they didn't have any of that. They had to sort of feel their way. It's like, well, what does this do? We don't know. We know the amino acid sequence of this protein. We know, but we know nothing about what it's doing. And so they had to map all that out.
And so then it's like like exactly doing the cell thing where you remove a cell and see what happens, but then you go one layer deeper and say, well, we have the whole genome mapped, so we have the ATGC sequence. And then if you can start to identify, well, this looks like a gene, this doesn't look like a gene, based on, you know, we don't have to go too much into the details, but there's little signals at the beginning and end of genes, conserved bits of A's and T's that let you know this is likely where it starts, this is likely where it ends. And you look for what happens if we change, you know, some A's and T's and C's in there? How does that then affect this ongoing development? So it's the same process, really, but again, you're just, because you have this, you can sequence the whole thing, you can get that data, and then start going. And then I think the other interesting point is, like you said, you can take genes from other organisms and compare them to what you have in C. elegans and say, okay, that looks similar, so maybe it does the same thing. And I guess that's the other big point about C. elegans, is that the stuff that goes on there is sort of like fundamental life, fundamental biology. It happens in every organism. So obviously humans are more complex, but the roots of it all are there in the nematode.
Yes. Yeah, I mean, I think one thing that I say to my students, because I teach some of this stuff to the genetics students, and one thing that's important to emphasize is a lot of this work was when this work to understand, say the development of C. elegans was being pioneered by Bob Horvitz, realistically, I suppose, at the early genetic screens. It wasn't a given that this would illuminate anything about biology other than worm biology. I mean, I think there was a reasonable bet, because of the way evolution works, that you would discover things that were important in all animals by studying the worm, but it wasn't a given. You couldn't say for certain, and I don't think anybody would have been able to say what proportion of the biology of the worm will also be true of the biology of humans. And so research councils in the US and in the UK were investing millions in this work, with not necessarily a certainty that you'll find something. No, it turns out some of the first genes that start falling out the screen turn out to be things like one of the most mutated genes in cancer, right? So Bob Horvitz was interested in understanding how a set of 6 cells go on to make a hole in the side of the worm, and it's the hole that's needed for the eggs to pass out. Okay, it's a very simple system. And they did something, we don't need to go into the details, but he developed a whole series of mutants where this didn't work properly. So the cells were developing wrong. And it turns out the pathway that they were using to become what we call vulval cells, the pathway they were using is exactly the same pathway that goes wrong in human cancer. Wow. It's the Ras tyrosine kinase pathway. And all of these components were identified and worked out first in the worm and in the fly eye, which is another nice thing. So Bob Horvitz and his co-workers were studying the worm vulva. A whole load of other fly researchers were studying how the fly eye is built during development. It's the same pathway being used, and it's the pathway that's mutated in human cancer. So, it's that kind of thing that I always say to the students, you should have chills going down the back of your neck when this is unbelievably powerful.
It's incredible. And it's like, and this is what when I talk to people about science and like, I really like to hammer on kind of the methodology of it, right? Like understanding the scientific method and that it's a process of discovery and that there's so much serendipity kind of involved in these things. And that's, the ivermectin story is filled with that. And this this nematode, this other nematode story is filled with that too, where you kind of taking a gamble as to whether this is going to work. Like, can we bring it into the lab, and all of these ideas that we might have. Okay, let's look at the fundamental biology of it, and then you see
Yeah.
that it's applicable here, and it's applicable here, and it's become this giant thing. So it's a very, very science story.
Yeah, no, absolutely, and I think I mean, the researchers that were involved in this, they were motivated by understanding basic biological questions, but obviously the people funding this are motivated by other things. It's like, well, if you're a research council, if you're a government research council, you're funded by the taxpayer. You can't spend millions say, well, this worm's kind of interesting. Let's find out about it. The taxpayers are going to rightly say, wait, what do we get out of this?
No, definitely. So then there's a few other key discoveries, and I think, maybe we can talk. So we have the laser that blasts out the cell, to sort of look at that fundamental cellular biology.
Yeah.
So then when we're talking about genes and things like that, there's a couple ways you can go about it, right? So gene knockouts or mutating the genes and then seeing what happens.
Yeah,
So this is a whole nother level of techniques that, were probably, really developed in C. elegans, other organisms as well, but they've become like these really useful techniques for all of these other things. So how exactly would you mutating a gene? I kind of get you can blast it with radiation, or maybe there's the drugs or something that you could put at during the development that messes up the genes. But like, we're talking about taking a gene. I'm assuming you don't use a laser to blast it out.
No, so this is actually an interesting thing. So all of the work I've been talking about was all about what we call classical genetic screens, where you're not targeting in a gene, and in fact you couldn't. When all of this was done, there was no way of sequencing a genome. Nobody had done any sequencing at that point, DNA sequencing. But then when I joined the worm community, I actually started working on parasitic nematodes. My PhD was working on a parasitic nematode, and parasitic nematodes are fascinating, but boy, they're hard to work with experimentally. And so I wanted to work on something that was experimentally a lot easier, which is why I switched when I finished my PhD to working on C. elegans. And at that time, we were just starting to get, well, the genome project for worms was getting underway. We didn't have a genome sequence, but it was becoming clear people were identifying genes that had no known function. And as we got to the stage where we've got the entire genetic code, we can then just look at it and say, look, there's a gene, there's a gene, there's a gene. What does it do? We don't know. And one of the projects that I wanted to develop for my postdoctoral research was a technique for just knocking genes out, and it didn't work. There was a lot of frustration in the field. A lot of the techniques that worked in, for instance, yeast, which is always the organism, brewer's yeast, Saccharomyces cerevisiae, we couldn't get them to work in worms. And it wasn't really until Craig Mello and Andy Fire systematically set out to effectively follow some weird observations that people had made, because that's where that work started. It was like, this is weird. I'm not going to go into we don't need to go into the details, but basically a set of researchers were doing what they thought was a particular gene knockout technique. But they're controls. So the experimental controls they did to show that what they thought was working the way it did, they didn't work. The controls looked bizarre. And so Craig Mello and Andy Fire systematically set out to go, there's something screwy going on here, but it might be important. And they were right, because they were smart people, and they discovered this phenomenon which we call RNA interference. Which I think was one of these left field things that when it was first reported, it was like, what? Nobody ever expected that to be a thing. And it was the advantage of RNA interference. So this is just the introduction of a particular configuration of nucleic acid. So we're all familiar with DNA. Possibly some of us are familiar with RNA, which is the sort of the cousin of DNA, and is often used to do does the work. If DNA is the storage, RNA is the RAM. Maybe, I don't know. I mean,
I think a way to explain it is DNA from genes gets translated or turned into bits of RNA that can then float around the cell.
So it's more portable.
Be turned into protein. It's like an intermediate between like you said, like the story or the words of DNA into a functional thing.
But what Craig Mello and Andy Fire showed was RNA had this other function, and in this other function that it had, it could switch off genes when it's given in a certain configuration, which meant that we now had a technique we could switch off any gene that we wanted, within reason. There's always drawbacks to them. There's always sort of caveats to these techniques, but and this is the reason why they won the Nobel Prize, because it turns out that it's not just worms, and it led to a whole. There are now therapeutics being developed for humans based effectively on that sort of technology. So these sorts of things were worked out in the worm, partly because, again, it's a simple system. You can do experiments on the fly quite quickly, which you can't really do in other systems.
And I think the RNA interference thing is a cool thing because, again, it shows that there's like this fundamental biology thing, this ability of RNA to, in these certain configurations, kind of just turn a gene on or off. And the body obviously uses that to help decide, well, I need a bit more of this gene product, or we need to turn this one off at this point, so we don't get tumor cells and runaway growth and all that stuff. Again, fundamental thing about biology. But then you can say, we can use this experimentally to turn genes off and see what's going on. And then maybe also, now with therapeutics and stuff. So again, another example in this thread I keep, I think, hammering home. But that's really interesting. And I want to then, I don't know, okay, we'll shift a little bit here. So we have this great model system, works great in the lab, and you were doing all these discoveries and stuff with it. But these worms, like you said, there is an isolate from some mushrooms that Brenner got, and those have now grown in the lab, and people will take a little bit of those and bring them to another lab or something. So you get lab cultures, and these every 300 or every 3 days, 300 more, but they're growing in conditions that aren't their natural conditions. And this is something that, again, you sent me in some of the papers, but while I was prepping for this, and I think it's an important point. And I'm viewing it from what we're learning now about genetics. It's not just one gene equals one function. The genes work together to do different things, so that's a level of complexity that you have to take into account. But also gene and environment interactions, right? That maybe there's some genes we don't know what they're doing, but that's because you're growing it in this sort of sterile kind of environment in agar, and maybe their function has something to do with the environment that it would encounter in its natural light. And so now the point is that they're kind of going back to nature to say, well, let's look at where this thing actually lives. And there's some kind of frontiers there, some questions still to be answered.
I think that touches upon an area as well that is a general issue for people studying laboratory organisms, but at least from the genetic standpoint. So one thing we haven't touched upon is C. elegans has the same number of genes approximately as humans. Now, when I say genes, I mean, I should qualify. So genes can make RNA, and genes can make proteins, but proteins effectively are the majority of the machines that build us. Enzymes are proteins for the most part. And so when we talk about 20,000 genes, we mean protein coding genes.
Okay.
There's an indeterminate number of genes that make RNAs. That's a whole separate story, I suppose. But let's just stick with protein coding genes for now. So worms and humans have about the same number of genes, which is in itself kind of an intriguing outcome. But one of the things that, if we study experimental systems, one of the things that we come across frequently as geneticists is that for many genes, when we remove their function, so we've got tools now where we can just take the gene out, it's gone. In many cases, the worm is fine. You know, it's happily doing its thing it's producing offspring. And so then that leads us to the question of, well, what is that gene is clearly important because it's there. It's been there for millions of years. It's doing something, but
evolution has kept it there. Yeah,
right. It's been maintained, but we don't know what it's doing. And a very distinguished developmental biologist, Lewis Wolpert, allegedly, I don't know whether this is true. I never, but on being told, this is about mice now. Are being told that this particular mouse gene knockout, so removing a gene from a particular mouse, the mouse seems fine for all of the tests. And Lewis Wolpert allegedly said, "Yeah, but did you take it to the opera and did it enjoy it?" That obviously that's a facetious comment, but it captures the idea of, we can't say that the gene has no function. All we can say is that in the pristine and incredibly coddled laboratory environment that we're raising these things, yeah, the gene has no function.
Right.
But why don't you challenge the organism with starvation or with a pathogen? I mean, one of the things that we're starting to discover is a whole load of natural pathogens that infect C. elegans, ranging from viruses, bacterial infections, and fungal infections. So there's a whole load of fungi that predate upon C. elegans in its natural environment, and presumably C. elegans has evolved strategies. And there are now research groups that are sort of co-raising worms and fungi, worms and their predators, to try and sort of tease out some of the interactions. And I think that's the thing. Our simple environment, we're missing biology, I suppose. But by growing them conveniently for us, we're missing out some of the biology which is clearly encoded in their genomes because of the selection pressure they experienced when they were, for instance, living in mushroom compost.
Right. Yeah, I think that's a really important point about the model organism thing, because there's so many, I think people often, there's a lot of things about model organisms that people maybe misunderstand or have problems with, but oftentimes I think in science, media or journalism or something, these important details kind of get lost, right? You hear about the discovery in mice that, well, we found this thing and it's going to lead to something great, and then as you move from mice to, say, another organism, say to human, the whole thing falls apart, right? And you might not know exactly why. And so that distinction, I think, is really important. And it's cool to me that, again, we have a great system in the nematode to go back to nature and start to look at some of these things. Because who knows what kind of, discoveries about immunity and, symbiosis and all of these different interactions, we'll find. I'm assuming you could even find something about microbiome with nematodes.
Well, people are doing that. There are more and more papers coming out around that area. Yeah, no, that's right. I think one of the advantages of C. elegans, as long as your question is about basic biology of being an animal, you could find a use for it. I work at a medical school, and early on, when I came here, I gave a general talk about what C. elegans could do for you. And one of the first questions I got was, well, does C. elegans have a liver? Well, not exactly, no. Does it have kidneys? No. But blood? No. And you could see all of the clinicians going, well, this is not even an animal then. How is that a basic, but I think in terms of, the key thing about animals is that they're multicellular, and cells are organized into tissues. And C. elegans does all of that. So if your question is related to that,
Then there it is. Then you have something. All right. Well, then let me shift gears a little bit, because I kind of touched on it there with, not the science media and the way that some of these things are reported, and that we're kind of lacking some of these details maybe that would, I think, provide a nuance to these sort of stories about discovery and whatnot. And I think that it's something that people who have listened to my show, I've said it a million times. I think we're really kind of failing a little bit on talking about some of those more basic concepts that we tend to gloss over. But it would give people a better understanding. And where I'm going is just basically to science communication, because I have you here, so I have to talk about it. And you sent me, and this was actually one of the things that John Gilliard mentioned when he said you should have Jonathan on the show. He was like, he even does some kind of stand-up, like some kind of science stand-up comedy or whatever. And so you sent me the YouTube clip, and it's great. So generally we can maybe move to this, because I think not a lot of researchers take the time to do science communication. It's generally unpaid, which is an issue. There's no, we don't have the mechanism where you can, when you're going for your raise or your tenure, to say, oh, well, I did X amount of science communication, so that counts for something. But I'll just start with, what is your motivation for doing it? Because you seem to enjoy it, and the YouTube video was great.
Well, I guess like a lot of researchers, or like a lot of people that go on to have university careers, your original motivation is the research. But obviously I think it's a cliche that you have people that they're in it for the research, but they know they have to teach because that's what pays the bills. And actually, I found that I really, really enjoyed the teaching, and I probably spent too long preparing my first lectures, because I found it fascinating. How am I going to get this rather complex set of facts across, or this concept across? And I really enjoyed that. And so public engagement was kind of a natural. I had a couple of friends who suggested for the science stand-up that maybe I could give it a try. I should point out, this is not something that this is not my own initiative. I've done it as part of a Bright Club. So Bright Club, I think, is still only a UK thing. It was started by Steve Cross at University College London. And it's now spread. Most large universities will have a Bright Club associated with them. And basically you get a proper stand-up comic who will train you to do a stand-up routine based upon your science. And it's frivolous as well. It is enjoyable. It's scary the first time you do it.
I can imagine.
But one thing it does do is it makes your communication ability but it improves that ability, because I found that forcing yourself to think about how to present a particular topic in an engaging and enjoyable way actually just changes the way you think. I guarantee that my grants, the grants that I have to write to support my research, I'm pretty confident that they've been made better as a result of doing science communication, because you're able to cut to the chase more clearly. You're more able to see, well, what are the important things that I need to get across? Rather than getting bogged down in the detail. And I think that's, for me, it's an unexpected trade-off. I wanted to do this anyway because I'm an absolute science nerd. And so I love reading about science. I love doing science, and I love talking about it. And so it was just another opportunity for me to do that. So I embraced that. But it was an unexpected benefit to say, oh yeah, okay, but if you're you can't use any of the jargon or the shorthand if you're talking to a general audience. So you have to think of other ways of doing it, and it's a challenge, but at the same time it also develops these skills that you didn't know might be necessary.
And I mean, I think storytelling is a big angle of that, right? Like there's also like you said, this cutting to the chase. What is the important details? How do I take something that is quite complex? And that's why we use jargon, right? Because you can say DNA translation or DNA transcription, and everyone who studies DNA knows what that is. But it's a step-by-step process of things that happen, right? It's not just two words. So you get that, but then it's also, how do I make this relatable? How do I make people
Yes.
excited about it in a way
Yeah,
that
why
they
should they
wouldn't?
care?
Yeah, exactly. And I think that's a really important thing that maybe, we're not, well, nobody gets training in science communication in academia, right? Maybe it's starting a bit
No.
more now, but you kind of have to
Yeah.
seek out those opportunities. You kind of have to show that you're interested in it before you can join the Bright Club or something like that. It's not like a mandatory part, but I think that would be really great because, like you said, it allows you to reflect on your own story of why you're doing this and the motivation behind it, which is good to tell the taxpayers, as you kind of alluded to before. But yeah, I think that's a big angle for it.
Yeah, yeah. And I think there certainly for genetics, there is a need to make sure that decisions that are made in, say, in policy outside of science. So genetics impacts areas of law now, for instance. So people have mounted murder defenses on the basis that they're predisposed.
Ooh.
They claim to be predisposed,
Right.
and having a scientifically literate populace to say, really?
Yeah.
That they're able to interpret that evidence is kind of important. There are areas in education. Do we test people? Do we perform genetic tests on people to see whether they're likely to have difficulties in education, and how reliable are those tests?
Right.
Spoiler: those tests are not very reliable.
Right.
That sort of thing. Genetics is leaking outside of academia, and it's kind of important. Therefore, that we-
This actually, so I didn't kind of expect to go there, but that's a really important point, I think, because of recent events in the US with some of the shootings that have happened there. There's a couple of researchers that I've noticed on Twitter that I didn't realize it, but going back several years, they have been talking about this misinterpretation of genetics research for nefarious reasons to kind of make these essentially eugenics right, arguments, right? These racial.
Yeah, that's another area as well.
And so,
yes, exactly.
being able, like predisposing people to genetics and how it works and stuff so that when they see those claims that are like dubious, to say the least, and misrepresenting the data or not showing all the nuance of or caveats, like you said, that these tests really aren't reliable in some way, is really, really important. It now becomes essential, right? That researchers stand up and start to say, no, that's not how this works. Let me clarify this. So because I've run into researchers that have said straight up, why does it matter if people understand what we're doing? Which I always thought was like a crazy thing to say, but now we have kind of a tangible example. So I'm glad you mentioned that, and not even just with this stuff, but the ethics of what we do with this information, how does it come into practice and stuff. It's really, really fascinating, and it's something that's going to probably impact our lives more and more as we move forward with CRISPR and different techniques and all this
exactly.
stuff. Yeah. So I'm going to go out on a limb here and say you recommend science communication to students, to your colleagues and stuff like that as not just an essential tool, but something that you can use for your own research.
Yeah, absolutely. I run an annual workshop for junior scientists to learn science communication skills. So I would definitely advocate this. Science is a community effort. I think unless you can communicate, I don't think you can actually be doing science. Now, because obviously within the team you're communicating, and it's I think it should be part of the standard set of tools that you should be mastering as a scientist. I'd say is that communication skills.
Yeah, because it's something that in my time in academia, and thankfully I didn't experience it personally, but you would hear stories or you would see really great researchers, at the pinnacle of their leading departments, they're doing those terrible communicators and not good at communicating with students, with colleagues, all of these things. And maybe it's the environment of incentives for getting grants and money and stuff like that kind of selects at some point for maybe a bit asocial kind of people that are really driven to do the research and stuff, really brilliant. But you need both, right? And so I'm wondering, what I'm getting at is maybe like, what were your thoughts on changing the system in a way to sort of encourage science communication? So is it mandatory courses? Is it, getting evaluated on your science communication, getting paid to do it?
I mean, I wouldn't want to make it mandatory, but what would be good, I think, would be is to increase the number of opportunities, because I think people will take them if they're given them. And you wouldn't, like you say, there's a place for everybody, really. Science should be about diversity. So you wouldn't want to say, although I've said science, part of your skill as a scientist should be communication. It doesn't mean to say that you should be able to communicate with everybody. And there has to be room for all sorts of people doing science.
Right.
But
not everybody's going to be good at it.
Mandatory. But for those people, but making opportunities and providing situations where you can be rewarded for those sorts of areas would be good. And I think universities, certainly I see in the UK, universities are changing that way. They're recognizing the importance of having public engagement.
Yeah. So, I think the UK, from my vantage point, seems like the leader in a lot of this stuff. You have what you're mentioning, these Bright clubs.
Bright Club.
But you also have, science festivals I've seen in the UK, things like this, where it's really, and we're just with the BBC and all the stuff that they do, they're kind of a leader in sort of wildlife documentaries or science documentaries, podcasts, all this stuff. So I think it's, obviously it's not perfect, but it's a real good, it's nice to see all that. And again, from a vantage point of outside the UK, an English speaking guy working in Germany, the UK seems like the Mecca in a way for science communication stuff. And I'll throw this last thought at you. I feel sometimes that, these things like the science documentaries, the BBC programs, not to disparage or anything like that, but a lot of things tend to, you find yourself in a formula of doing this science communication, how to write the news pieces, the documentaries, and it all starts to kind of sound the same. So I'm wondering about your thoughts on how do we, diversify that? How do we make that a bit more? And I think the stand-up thing is a great example of that, because I worry that a lot of science communication, all of this effort that we're putting into it, ends up speaking to the people who are already seeking it out. How do you break out of that? Is it going into schools and starting at a younger age, or what do you think?
Yeah, that's
an unanswerable question for you, but
that is the perennial problem. Yeah, that if you have a science festival, the people that come will be the sorts of people that go to science festivals. Yeah, I don't know what the answer to that is. One of the initiatives that we have in Scotland is researchers that go and do public engagement in prisons. So environments that typically don't have access to this. But actually, as it turns out, I've not done it myself personally, but I've got colleagues that have done it. These people are hungry for this sort of environment, for these sorts of interactions. So, I mean, that's one way, I guess. That's the perennial problem. And again, the other thing, public engagement has its own set of professionals now who think about these problems. I'm certainly not. I'm an absolute amateur. But there are teams now that research this problem of how do you go about engaging people that probably wouldn't think of themselves as the audience, because that's one of the things. Science has a diversity problem, right? So this is self-serving. We know this. It's not a UK problem. It's a global problem. We have an issue with the sorts of people that get recruited into doing science. Some of it's unconscious bias, but some of it's sort of structural things that are there that prevent us from having a truly. I mean, not to get all Carl Sagan about it, but I mean. It's a uniquely human enterprise. So science should reflect the fact that it's a uniquely human enterprise, and so we should see that diversity. And we don't. I mean, it's clear we don't.
And I guess that's like I kind of alluded to, sort of incentive structures that select for different, things that, allow a certain individual or a certain type of individual to succeed. And then that's really what we're talking about, I think. But it is a good point that we talk about serendipity and all of these. You never know where the discovery is going to lead you, right? So having a diversity of opinions of people with different life experiences who have come from different environments and stuff like that, they will view problems differently, and they will see things that the other person. And so it really is a community effort. There's no one great scientist that's working alone and discovering all this stuff, right? Like, so to have that back and forth, to have that as many ideas in the room kind of thing is so important. So, and I think you kind of, let's say this is a takeaway, is bringing the science communication to audiences that aren't, not asking them to come to the festival, because you're going to get all the people that want to come to the festival, but how do we put it in front of audiences that weren't expecting it, kind of thing? And then that comes
to don't see themselves as scientists.
Right, exactly. Right. Because it does touch their life, whether they know it or not, and they probably are scientists in some way, in the way that we all figure things out. That method of learning is science, essentially. So how do you package it in a way that maybe it's not quite the right way to put it, but slip it in without them knowing it? So maybe they're going to the heavy metal festival, but there's some kind of some kind of science there. I don't know, but But I'll say this has been great. I really appreciate you taking the time. And
I've really enjoyed it.
Yeah, and there's so much more we could talk about with C. elegans. So maybe we'll have to connect again in the future. But I'll just leave this. I always ask everybody: is there something that we didn't touch on, or that we kind of glossed over, that you think is important to add?
No, I don't think so. I mean, I could probably talk about C. elegans for days. Specific minutiae that are absolutely fascinating. But I think we've covered fairly well the sort of broad picture of C. elegans as a research model. Yeah,
we definitely hit the highlights in my mind of just, wow, so much knowledge has come out of this one tiny worm that some guy had the foresight to see, rotting compost, little worm, to understanding life. It's a pretty big leap, but pretty amazing. So thank you so much, Jonathan, for doing this
thank you.
Once again, and good luck in the research, and hopefully we'll talk again in the future.
That'd be great. Thanks very much.
There you have it. What a story. What a story of discovery. Didn't realize we were going to be stepping into a fruit fly versus nematode debate struggle there, but I'm glad we did. Nematodes all the way. You know what team I'm on. And many thanks to Jonathan Pettit for joining again and sharing these stories and sharing his experience with science communication and his work with the nematode C. elegans. It was a great conversation. I really enjoyed it. So thank you, Jonathan. And again, as always, please rate, subscribe, tell a friend about the show. Visit the website, twobradforyou.wordpress.com. Get in touch with us. We'd love to hear from you. And please, if you feel like it, donate again at the website, twobradforyou.wordpress.com. You can find it all there. Thanks, everyone. Have a great summer. Bye for now.